The overall goal of this workflow is to determine whether foci identified within the cytoplasm are bona fide stress granules, which are RNA granules that play important roles in cell physiology.
The main advantage of this workflow is that it tests for all the fundamental properties of stress granules. Though this workflow is presented for human osteosarcoma U2OS cells, it can also be applied to other types of cell lines and primary cells. Generally, individuals new to this method will struggle because not all stress-induced foci are stress granules. Our workflow comprises all experimentation necessary to distinguish them. I will be demonstrating this procedure with Anaïs Aulas. We are postdoctoral fellows in the lab of Paul Anderson and Pavel Ivanov.
To begin, seed cells onto cover slips placed in 24 well plates as described in the text protocol. After overnight incubation, aspirate the culture media from wells B1 to B4 and from C1 to C4 of the 24 well plate. Add 500 microliters of the medium supplemented with 100 micromolar sodium arsenite to wells B1 and B2. Then, add 500 microliters of the medium supplemented with 50 micromolar sodium arsenite to wells B3 and B4. Next, add 500 microliters of the medium containing 150 micromolar vinorelbine to wells C1 and C2. Finally, add 500 microliters of the medium supplemented with 125 micromolar vinorelbine to wells C3 and C4. Incubate the plate for 30 minutes at 37 degrees Celsius. After 30 minutes of incubation, add to the cells in column 2 5 microliters of 1 mg per milliliter cyclo